How did the unique membrane we see in bacterial species like Mycobacterium tuberculosis and Mycobacterium leprae originate? Was it from a gram-positive or gram-negative ancestor? Or could they represent the last descendants of an even more primitive or less derived clade of bacteria? The Mycobacteriales order belongs to the actinobacteria phylum, which is predominantly gram-positive with a few exceptions mixed in, as well as, of course, the gram-indeterminate Mycobacteria. One phylogenetic analysis from 2018 points to Mycobacteriales as a derived clade within the actinobacteria. So, the membrane of Mycobacteria almost certainly do not represent some sort of primitive state. So, Mycobacteria are derived clade. Derived from what? A gram-positive monoderm ancestor or a gram-negative diderm ancestor? Alright, case 1, a gram-positive monoderm ancestor. Pretty straightforward. Take a monoderm, add a membrane to it, much like might have happened in producing the first gram-negative bacteria, depending on what theory of that you subscribe to. Because if it happened once, why not a second time? And this could explain the wildly different architecture of the mycobacterial membrane compared with other diderms, the gram-negative bacteria. But if you think about it, is a gram-negative diderm ancestor not a more reasoned theory? Adding a whole outer membrane is no mean feat. We have to jump through a whole lot of hoops to add another membrane to a bacteria. You need both leaflets of the outer membrane itself, as well as something to anchor it to the cell wall. And it's really hard to come up with plausible intermediates along the way. One of the most popular theories for the acquisition of didermy in general ties it to endosporulation, which, if real, is a pretty cool example of exaptation, I think. But is it not easier to start from a gram-negative diderm, change out some components one at a time, and end up with your mycobacterial membrane? This is honestly one of those things we'll never really know. If any intermediates in the evolutionary transmission ever did exist, they certainly appear not to anymore. And while some artificial evolution experiments could play back the course of evolution and show that one pathway or another is plausible, demonstrating something in a lab doesn't show that it's what necessarily happened in evolutionary history. But speaking of artificial evolution, there was a cool recent experiment investigating how mycobacteria acquired pathogenicity in mammalian hosts. So I'll bring that to you in a forthcoming video. But that might be a while, because maybe gonna take a little break from talking about bacterial membranes for a while. See ya.